Okay, we're recording. All right. Okay, well, let's take a look now at the sperm's response, or rather the egg's response to the sperm. We described before, the sperm come in when it contacts the appropriate receptors, which are usually embedded in things like the vitelline membrane, egg jelly, if there is egg jelly in that type of egg. And that triggers off events, especially the acrosomal reaction, where we release the contents of the acrosome, which are full of enzymes that basically drill a hole through all that material so that the sperm can swim into the egg and actually contact the egg plasma membrane with that second set of receptors, which are hidden in the acrosome vesicle wall. And of course, they're exposed when the acrosome secretes. Okay, now, the egg's got to respond to the sperm, and we take a look at the egg. Okay, the plasma membrane over here, just beneath it, and we're talking about a fraction of a micrometer, are these cortical vesicles. They're filled with enzymes and other contents. They're just beneath the surface of the plasma membrane. And of course, under the right event, they're going to trigger off and simultaneously all secrete their contents, and that plays an important role. And then, of course, we have the vitelline membrane. And it's connected by proteins to the plasma membrane, so it's really close. And there are receptors that stretch through the vitelline membrane that will pick up the sperm, and sometimes a second set of receptors further on. Okay. Now, when we get this sperm-egg interaction, what's going to happen is it will trigger off the activation of the egg and what's called blocks to polyspermy. Now, polyspermy means many sperm fertilizing the same egg at the same time. And when that happens, of course, instead of having 2 sets of haploid chromosomes, you're going to have 3 or 4 or 5 or whatever. It's a developmental disaster. I mean, development quickly arrests itself. So you want to block polyspermy. And we'll see there's 2 ways of doing it. One happens within seconds, what we call the fast block. The other happens in half a minute to a few minutes, and that's called the slow block. And that includes with the so-called cortical reaction and formation of the fertilization membrane, which we'll see in a little bit. Okay, now, there's various kinds of receptors and stuff. When you're looking at mammalian sperm, mammalian eggs, of course we have the oocyte is surrounded by layers of follicle cells, which are right over here. The follicle cells also have receptors on them that will bind the sperm. As a matter of fact, in mammals and in humans, we identify that receptor. Okay, and it's called ZP3. It stands for zona pellucida 3. Now, zona pellucida is a fancy Latinized name, this is for the medical students, for the vitelline membrane, because, of course, it's a glycoprotein that extends into the vitelline membrane. Follicle cells also have ZP3 on them, too. So when the sperm contacts that, that's going to trigger off the acrosomal reaction in the sperm, and it's also going to start triggering off events in the egg. Now, the interesting thing about ZP3, since we identified it, I believe we know the tertiary structure of that protein. You can think, oh boy, if we could block ZP3, we'd have a great contraceptive that's not a hormonal-based one. There's already been some experiments done. At first, somebody tried the idea of what happens, and they did this with mice, which have their own version of ZP3. The idea of basically taking ZP3 and injecting into the mouse and basically immunizing the mouse against ZP3. It worked. The problem is it was also irreversible. It was one of those kind of things like you get the smallpox, or you used to get the smallpox vaccine and basically lasts for life. Or if you get, the chickenpox vaccine that basically is a long-term or lifetime vaccination. So it didn't work. It wasn't going to be suitable for people if they wanted to later on have children and stuff. But there are some companies, in fact I think especially Chinese companies, that are working on blockers, stable blocking small molecules that could go in, bind to ZP3, and basically when the sperm came in they wouldn't have anything to bind to. That would be a block to fertilization. It would, if it pans out, it's non-hormonal, so it's not going to have all these other potential side effects that things like the pill and Depo-Provera and stuff might have. For people that believe that a fertilized egg counts as a human being and has the same rights as a human being, that eliminates the issue of are you destroying a person in the form of a fertilized egg. So there's several different pluses on that if this actually pans out. So it's hard to say, but people are doing work on that as a potential contraceptive that would be very specific, much safer, and eliminate some potential ethical issues. Yes?
Did they not have someone with them produce the antibody?
Well, problem is trust me, I know this. I know this firsthand. You react to foreign antibodies. They're recognized as foreign and they can cause some nasty side effects. I had a few kind of, fortunately, short-term effects from the first time I was getting antibody infusion because they're basically mouse antibodies. So I started squeaking, I was looking around for some cheese and stuff like that. Okay, but anyway, yeah, so that is a problem, injecting foreign antibodies. And you can mount a nasty and sometimes even fatal immune reaction to foreign antibodies. Okay, but anyway, so people are working on the potential idea, can we block CP3 and in a reasonably long-term but not permanent fashion, and it'd be nice if you take a pill once every week or something like that, and that protects you from pregnancies that you don't want. Yeah, you had a question?
Yeah, so ZP3 is found on follicle cells only?
No, it's follicle cells and goes through the vitelline membrane, ZP, zona pellucida, aka the vitelline membrane of mammalian and human cells.
So those receptors are on both?
Yeah.
Types of cells?
Yeah, exactly. Okay, well, of course, the sperm comes, it's going to hit the follicle cell ZP3, fire off the acrosomal reaction, and tunnel right through, here, and then bind to the egg's plasma membrane. Okay, meanwhile, that's going to be triggering off a number of events in the egg. First thing, let's see what goes on first. First thing that's going to happen, for the most part, this uses probably also a tyrosine kinase involved that we're not quite sure exactly what it does, but there does seem to be a tyrosine protein kinase involved in some of this as well. But the main signaling pathway, once again, is our old friend, the phospholinositide pathway. So here we have the receptor. It activates a trimeric G protein. The alpha subunit falls off. It binds to phospholipase C and activates it. Phospholipase C takes the membrane phospholipid, sometimes called PIP2, splits it into two components, the IP3, the inositol triphosphate, causes calcium release from the inside, and then the diacylglycerol, which is very hydrophobic, activates protein kinase C. Okay, you're going to get a number of things happening from that. Several things are going to happen pretty rapidly. First of all, you're going to get a lot of changes in ions. We're going to activate a couple of different kinds of things. we're going to get release of calcium from internal storage, mostly the ER. The mitochondria actually store calcium too. So calcium is going to be released from the inside and go into the cytoplasm. Simultaneously with that, thanks to protein kinase C, we're going to also activate, facilitate diffusion calcium channels in the plasma membrane. So we also get a calcium ion influx through channels. So calcium is rushing in from the outside and being released from the inside. Now, for the most part, the major source of the calcium is actually from the inside, but external calcium coming in plays a significant role as well. So calcium is flooding in, and calcium is being released from the inside. And of course, we'll see what calcium ions do shortly. A second thing we're going to do, once again, is we're going to activate a sodium ion H+ co-transport system in the plasma membrane. What that means the inside of the cell has a lot less sodium than the outside of the cell. So sodium rushes into the cell by facilitating diffusion, and the energy from that is used to actively transport H+, hydrogen ions, out of the cell. And that causes an internal pH rise, generally from about 6.8 to about 7.2 or 7.4 or so.
Does this calcium influx, like, directly rise to the sodium?
No, actually the diacylglycerol activating protein kinase C does that. The
Okay.
PKC phosphorylates those calcium channels on the outside. Now the internal calcium channels, those are opened up by binding IP3. So here you have a ligand-gated calcium channel on the inside, IP3 opens that up. And then you have a calcium channels on the plasma membrane, they're regulated by phosphorylation, at least at the minimum. And those open up. Okay, so we're seeing massive changes in ions. The cytoplasm is getting flooded with calcium ions. And recall, calcium does all kinds of things in cells. We'll see what in a little bit. We're also raising the internal pH of the cell. Now here again, anybody took cell biology with me and remembers that enzyme lab where he stuck the enzyme and measured its activity at various levels of pH? We could see that the activity would change depending on what pH it was exposed at. I promise that some batches of that darn dopa also tend to hydrolyze spontaneously at high pH, so that guy throws a monkey wrench. But most of the time, each enzyme, each thing's going to work well at some pH and not at others. So you do have pH-regulated proteins. Okay? So what we're going to do here, calcium is going to do all kinds of things in cells, including protein regulation, and we have a pH increase, and that's going to regulate numerous proteins too. So what we're finally doing, and these lead to what we sometimes can call egg activation. Okay, so that's our little bit of signal. Yes.
So, based on the C, the calcium 2+ that causes all that. Okay.
Okay, well the calcium, well these are just the ions that are coming in. Calcium releases all kinds of different things, right? Okay, and then a separate thing that happens is this co-transport, and that's actually courtesy of the protein kinase C phosphorylating those proteins.
Do they happen at the same time?
More or less. Probably the calcium probably takes a little bit longer. This seems to happen in a few seconds. This you start happening in tens of seconds to a few minutes. Okay, so there is a little bit of a difference in time here. Okay. Now, several things are going to happen. we're going to generate the polyspermy blocks. In other words, mechanisms to prevent more than one sperm from fertilizing the same egg. And then we're also going to do steps that activate the egg's developmental program. So we'll start the program of early development even before the two nuclei have fused. Okay, so let's take a look at some of these events. Well, the most important is blocking polyspermy. That is a very bad thing developmentally.
So actually